All right, thank you, Anne. So now we will open up for questions. Again, if you have questions you'd like me to ask Anne, please type them into the Q&A box. We'll start with a quick question about use of the website. So someone asked, if you get to the variant page, like from ClinVar or the allele registry, what's the best way to get back to the gene page from there?
Yeah, great. So I thought I would just share my screen and show you this. And one other feature I forgot to mention when I was showing you the website. So if you click from many other resources, you'll get to this variant page where you can see, again, all the information about the gene. There's also a question about the allele count versus the allele number. So that's the allele count is the number of people who have that variant, and the allele number is the number of people that we have genotypes, sorry, the number of chromosomes we have genotypes at that site. So 2 times the number of people for if the chromosome is not a sex chromosome. So that's really helpful. And we talked a little bit in the previous talk about coverage differences, and that's why this number can vary from variant to variant. If you want to look at the gene page from here, because after all, you have all this wonderful variant information, but I want to see the whole gene, you can actually just click on the gene name anywhere it's located on the screen. I don't think it's up at the top at all, but if you click on the gene name anywhere else on the screen, it'll take you to the full gene page. One of the things that I think is a really subtle feature here, but is really important, is that there's all these question marks around the page. So there's a question mark in the bottom right-hand corner. If you click on that, it gives you lots of information about how these different processes, like how the variant QC happens, all these different things. And then there's some hover overs with information, and then scattered throughout, again, lots of information, how constraint is calculated, how our transcripts are done, where they come from, what subsets mean. So tons of information just hidden in these little question marks. So I encourage you to check those out also.
All right, thank you. Now we have a couple of questions kind of surrounding a similar theme. So I'll start with the first one. How representative are the samples used to derive these rich genomic data? For instance, it's apparent Africans are only gradually becoming aware of genomic studies, and only a few are consenting to participate in these studies. Consequently, African population may not be well represented in these data sets. If that's true, how generalizable will the findings how generalizable are these findings?
Yeah, so the more diverse, the more inclusive of every population these reference databases are, the better resource they will be for individual communities, but for everyone. So seeing a variant that's common in Europeans is very helpful to East Asians and Africans. And so knowing a variant is common in any population is still very helpful to every population. But there are definitely populations that are less well represented. African is one of them, Middle Eastern is another, and Oceania is another region that's not well represented. But even within East Asian, you can see we break that ancestry group out because we have some of the different country information available. You can see like Japanese, for instance, has a handful of people in that data set versus in the gnomAD data set versus more from Korea. So in general, the more representation we can get from different communities, the absolute better this resource is going to be for everyone who uses it. But you're right, if you are African and you have genetic testing done, we all have variants that aren't in gnomAD. If you look at a European ancestry individual, they have about 10 variants that are absent from gnomAD, give or take plus or minus 10 probably. If you take someone of African ancestry, they might have more like 30 or 40 variants that are not present in gnomAD. And so that's more 30 or 40 variants that you don't really know much about that are going to be harder to sort out. Those aren't going to be 30 or 40 damaging variants. They're just variants that we don't have as much information about. So getting more people from different ancestry groups into gnomAD is going to help with that. Getting the way gnomAD is done is it's an aggregation study that brings together existing data sets. We don't sequence data just to be part of gnomAD. And so, but the data has to actually physically come over to the Broad Institute at this point, be processed through these same parallel pipelines, and then jointly called as a single unified gnomAD call set. So it's very high quality for the community. So we're not introducing sequencing artifacts into the data set. We wouldn't want to do that because then it could decrease its value for diagnostic testing. So there are efforts, and it is important to be able to bring in more diverse data sets. And so Dr. Heidi Ream, who's involved in this workshop, is now one of the PIs of the gnomAD project. And so she's a great person to talk to if you have data sets that you'd be interested in trying to contribute to gnomAD. There is some costs and definitely a lot of human effort involved in bringing data sets over, but it is an important thing. And so it's something that is definitely worth further exploration. We'll be working on in the coming years.
And I know, some of what you just said also partially answers an additional question: what could be done at the African level to increase diagnostic yield of genetic tests? But would you add anything additional from what you've already said?
So we've talked about sort of two sides of things. So the reference population data, but also increasing access to diagnostic testing and making sure that the results from diagnostic testing and research studies on disease populations are getting shared in resources like ClinVar, so that there's a growing catalog of the pathogenic variants in the African community is going to be really important also.
All right, next question came in twice. Are variations seen in a single genome the same for all populations? I assume that those from an African with Black ancestry may be higher.
So I think what you're getting at is the just sort of amount of variation in an individual, that in African individuals particularly versus European ancestry, but versus many other ancestries, there are more common variants and more rare variants. And that's one of the things we actually take into when we do the QC to make sure that the data sets look good, that we want the African samples to cluster with each other, but they do cluster separately from the European, and there are absolutely more variants, which is also a reason that having more African individuals in the data set really adds a lot of new variants that we have information about, and so are very powerful addition to the database.
Okay. Will there be any Southern African genomes included in gnomAD version 4?
I do not specifically know the answer to that. There's a lot of new data sets coming. The goal for version 4, which again we hope will be later this year, is hundreds of thousands of exomes. So there's a lot of new data coming in, and I don't off the top of my head know, but if you're interested, follow up with an email and we can look into it.
All right. And just to clarify, in the next version, again, version 4, will the exomes and genomes from previous versions all be included together?
So this is always a little point of somewhat confusion in that from EXAC to gnomAD version 2 to version 3, most of the samples are carried over. The QC pipelines are a little bit, like as the data set grows, sometimes some of the samples that are kind of borderline on QC might fall out. So it's not that 100% of the samples from earlier data sets are included, but like 98, 99% of the samples are included. For version 3 and version 2, there is a fair amount of sample overlap. Pretty much all the genomes in V2 are in V3, and there are some exomes that now have genomes or also have genomes that are now in V3. So you can't view them as independent data sets. If you're looking at data in V3, again, remember it's on a different build, but there's actually a subset that you can say, can I look at this variant but not in V2? So that you can try to differentiate how many people are actually in all of gnomAD with that variant.
All right, and then how about graph genome projects? How can we make use of them in the database?
So it's not really as much related to gnomAD, but there's a lot of genetic diversity that both at the single nucleotide level and at the copy variant level that is going to be important. And there's been some thought that building kind of personalized references is going to improve the ability to map and diagnose patients, particularly ones that aren't caught through kind of standard diagnostic pipelines. I think that's an interesting idea for future research and exploration. It's not something the gnomAD project is really involved in, but I'm definitely keeping my eyes and ears tuned to see what comes out of that.
All right, and in the last 5 minutes or so, when you have a variant that looks interesting, could you talk more about what additional steps that someone could take to figure out the importance of that variant?
So this is one of the big conundrums in our field. So I'm also a clinical geneticist, so I see patients, I send diagnostic testing, and I get these reports back with variants of uncertain significance. And it's hard. So you look at the ACMG criteria and think about the types of evidence that would help you increase the knowledge about the variant. But most often it's going to be something like trying to find some functional data. which can be challenging. I do sometimes email people who have published on a gene just to see if they'd be willing to, if they know anything about my variant, or if they're willing to do some additional work on my variant, or like my variant that I found in the patient through testing. And then the other thing is putting things, sometimes I'll even enter something into ClinVar as a variant of uncertain significance, just in the hope that other people will like look at that variant and start to share more information about it. Because the second approach really is waiting and watching and seeing when more information is available, and more information is typically more patients or more people identified with that variant, which can be frustrating, but there's a lot of variants, and so there's not always a good follow-up. I wish we had more easily accessible functional assays.